Yes.
Okay. So those who are outside, my name is Gabriela, and I'm working at the LSA facility together with Sylvie. And today I will talk about immunostaining, and what is this all about, and some tips in how to troubleshoot your immunostaining sample. So during this lecture, I will touch the following points. I will go through a very quick reminder of how the immunostaining works, what type of antibodies we are working with.
Gabi, do you mind hiding the panel at the top there, because it makes like one big black square? So on the, I think, right side somewhere, you can say hide. Like if you click on the three little dots or something, you hide the floating panel or something like this.
Yeah.
So it will disappear. Thank you.
We will go through in a immunostain, what is the difference between IHC, ICC, and IF. If there is a difference and why. If we immunostain the cells or tissues, is the same thing, or do we have to think about something different between them two? What's the difference between chromogenic versus fluorescent labeling? What kind of controls we should always have when we perform an immunostaining? How we choose an antibody? What is a good antibody? And I will again highlight some important some things, some parts within the important steps in immunostaining: fixation, permeabilization, antigen retrieval, blocking, and washing. I will make some suggestion and highlight some important ideas and a little bit of a conclusion. So let's get started. So how does immunostaining work? So antibodies are macromolecules that are naturally produced by animals to defend themselves against proteins that do not belong to them. And antibodies only recognize proteins, no other type of molecules. The protein that is recognized by an antibody is called antigen, and the part of the antigen that the antibody binds to is the epitope. So let's say here is a protein coming from a red human. When this purified protein is injected into a blue rabbit, the rabbit immune system will respond and it will produce antibodies against this exogenous antigen. This is not belong to him. The most antibodies come in different flavors called isotopes, and the one that is mostly used in our line of work is IgG. Mammals produce 5 different type of antibody isotope, and each of them have different subclasses. In our example, for example, we have the rabbit produce an IgG2A antibody, and where the IgG is the class and the 2A is the subclass. The IgG antibodies are composed of 2 identical pairs of protein, each made by a heavy chain and a light chain. And the 2 of them are assembled together, as you can see, in a Y-shaped structure. The antigen binding part is called the FIB fragment, and it's antigen specific, and it will be bind specifically in our example into the red epitope. And the FC fragment is the part that tells the rabbit that it has produced its IgG, so that it's recognized it as its own. To detect the antibodies, we have 2 options, either direct labeling, and in that case, the FC fragment of the IgG is labeled so it can be detected. And the second alternative is indirect labeling, where the FC fragment coming from the rabbit is injected in, for example, a green mouse, and the green mouse which will respond by producing its own IgGs with a mouse FC fragment and then FAB fragment that will bind the blue rabbit FC part. And then these mouse IgGs are labeled so they can be detected. The unlabeled antibody used to detect the antigen is called primary antibody, and the labeled mouse IgG is called secondary antibodies, and it will bind the rabbit primary antibody. Okay? So now let's briefly go through the types of antibodies that we are working with. So we have monoclonal and polyclonal antibodies. The polyclonal antibodies are produced by injecting the host animal with the antigen that will lead to an immune response. And the antibodies are then extracted from the animal blood. Usually, rabbit, sheep, goats are the most commonly used mammals to produce polyclonal antibodies. Chicken are also used, and they produce IgY antibodies, which are equivalent to mammalian IgGs and are very, let's say, conveniently harvested from the egg yolk. But remember that the polyclonal antibodies are a mixture of antibodies that all recognize different epitopes on the same antigen. And that would lead to a relative high batch-to-batch variability. And that, which also means a higher chance of cross-reactivity due to a recognition of multiple epitopes. Monoclonal antibodies are generated in vitro by a hybridoma cell line from mice, recognize only a single epitope on the antigen, and thus we will have a low batch-to-batch variability. But it takes time to produce. Recombinant antibodies are monoclonal antibodies that are produced in vitro by cloning the antibody heavy and light chain in expression hosts. Basically, you are cloning the antibody heavy and light chain DNA sequence from the B cells or plasma cells of immunized animals into these recombinant vectors. And then these vectors are introduced in expression hosts, for example, the E. coli, who they produce the antibodies. This technology provides an unlimited supply of identical antibody, removing the batch-to-batch variability. Other type of antibodies are FIB fragments, and usually they are generated by enzymatic digestion of the whole IgG antibodies. With papain digesters, you are removing the entire FC fragment, including the hinge region. They are monovalent, so they only have one antigen binding site. And I think that the molecular weight should be around some 50 kilodalton. And they can be used for blocking cross-reactivity. They're using primary antibody raised in the same host species. And the good thing about them, because they are much smaller, they penetrate tissue more efficiently. But they will give rise to a lower signal if fluorophore is attached to them, because there are fewer fluorophore enzymes than can be conjugated to them. Now, if we have another enzyme which we add to digest the IgG, pepsin, for example. This will also, this will only remove the FC part and will keep the FAB parts linked together by this hinge region. And they has two antigen binding site. They were usually used in special application, like for example, blocking binding of secondary antibodies to FC receptors. Now, besides the 5 conventional classes of mammalian antibodies, some antibodies have a completely different structures because they don't have a light chain. These antibodies are found in only a few types of animals. They were initially identified in camels. That's why they are called camelids antibodies. But they are also present in related species such as llamas, alpacas, and some, interestingly enough, some shark families. Now, this small variable part of these antibodies is homologous to the variable part of the IgG, and it can be isolated, and it's called nanobody. So the small size of the nanobodies and the fact that they are formed by one single protein offer some advantages. Like, for example, it can be easily cloned in expression vectors and transfected into the cells. Of course, also tagged with fluorescent proteins. And thus, it can also be visualized, easily visualized in living cells. So now that we know how what antibody we are working with and how they work, so let's talk about move on and talk about immunostaining and the different types. Or there is a difference or not? Let's see. Then we, when talking about immunostaining, one can use the word immunofluorescence, which is abbreviated IF, or immunohistochemistry, which is abbreviated IHC, or immunocytochemistry, abbreviated ICC. These terminologies, actually, they are all refer to immunostaining. So if you call them immunostaining, you will never go wrong. But let's see, let's break down down the words so we can remember a little bit easier their meaning. So the I comes from the abbreviation immuno, and just tells us that it's an antibody-based method to detect a specific protein. So that's why there is an I in all the three techniques: IF, IHC, and ICC. The H, it's a shortening from the histo, abbreviation of the histo, shortening, and it means tissue. So this type of immunostaining is done on tissues or 3D tissue models. The C, it's a shortening that comes from the cyto, which means cells. And so this type of immunostaining is done on cells, cell monolayers or cells in suspension. And the last letters are C or F, and basically correspond to chemistry or fluorescence. And they describe the two methods used to detect antibodies, either using an enzymatic reaction, chemical method, or fluorophore, which is the fluorophore method. Now, let's see if we are, now that we know what IHC, ICC, and IF stand for, let's see if we are talking about immunostaining of cells or tissue organoids, is there actually any difference? So then we have the immunohistochemistry and immunocytochemistry, differentiate, obviously, if the sample is a cell or a tissue, but not immunofluorescent. And this is only of historical reasons. In reality, regardless of the visualization method, if we have a chromogenic or a fluorescence-based visualization method, the difference between staining of cells or tissues lies essentially in the steps required before immunostaining. So, cells, monolayers, or cells in suspension that are cytospin are very thin samples. All you need is a simple fixation which is required before starting your immunostaining. So it goes fast and it's very easy to perform. Tissues, on the other hand, are much thicker, so additional processing steps are required. Now, depending on your scientific question, but you have how precious your sample is and what kind of microscope is available, one can decide to use either a whole mount, the whole tissue as it is, or to section section the tissues. Now, the whole mount processing includes always a fixation step, and usually this is done by perfusing the animal with a fixative. It also requires that the tissue is made transparent using different chemicals, and with the exception, of course, of the zebrafish, which is naturally transparent. And the chemical process that makes the sample transparent is called clearing. Now, the success of the light sheet microscopy has led to an explosion of different protocols for clearing for different tissues and different tissues, different fluorophores. So basically, all you need to do is to find the clearing protocol that works for you. So this is our recommendation: going out in the literature and find something that was already proven that it works for your type of tissue. And then you are imaging the whole mount. The advantage that you get, it's an excellent 3D knowledge of your tissue. Now, alternatively to process them as a wall mount, what you can do is to section your tissues, and there are two strategies that will give you the stiffness that is necessary for this tissue to have in order to be able to cut them. The first strategy is freezing the tissues, and the second is without freezing them. Now, if you are freezing, again, you can have you take your tissue and you freeze them at once, or you can first fix and then freeze. Fresh freezing means that you will embed your sample without any prior fixation step, and you will basically embed it in an OCT block, which then is quickly fresh frozen isopentane, which is cooled in liquid nitrogen. And now this OCT block is hard enough to be possible to cut your section. So whenever you need, you take it out, you are sectioning, and then you will fix your sample and start your immunostaining. So all these steps in this method, freezing, sectioning, and staining, are relatively easy and fast. But unfortunately, also this method, this is the method where we will get where the tissue structure has the greatest risk for being seriously damaged. In the second way is to first you will fix the tissue, then you will immerse them in a cryoprotectant like sucrose, which will prevent the water molecules from forming destructive crystals during the freezing process. So this actually will help quite a lot to preserve the tissue structure. Now, after that, you will immerse them again, will embed them in OCT, and you will froze in isopentane or dry ice. This method, and afterward, of course, you will section whenever you need. And this section is much slower than the previous one. But then it always also you could also probably get some tissue damage. The second way to produce to be able to produce samples for sectioning is without freezing, and in this case, you will first fix your tissue, and after which you will embed it in paraffin or low melting point agarose. The paraffin is a thick oily material, and this is used for tissues. On the other hand, agarose is water-based, and it's much softer. So it can only be used for very soft material like 3D tissue models where the cells are growing in a gel. After you embedded your samples, you can cut them in very thin sections. This the paraffin embedded sections and blocks can be stored for a very long time. There are unfortunately some disadvantages with this method. First of all, the procedure is much longer, and the paraffin embedded samples are quite often autofluorescence, and this process also sometimes kills the fluorescent proteins which are expressed as reporters in transgenic mice, for example. Now, when we are talking about fixing tissues, strong fixation is usually needed, and sometimes this will mask the epitope, so the antibody will have a hard time to reach and bind. That's why all these methods require antigen retrieval. So they are quite, which is quite harsh for the tissue, and quite often also will lead to the tissue detaching or be destroyed. So now we have an idea a little bit what's the difference when we are working with cells or when we are working with tissue. So we can assess and understand what we will have to do when we are deciding what kind of method we will use. But I would also like to highlight that so we know we will know what to ask for when you are cutting your tissues. There are unique special instruments that are needed, and for example, and different instruments will produce different a section of different thicknesses. So cryostats are always used with frozen tissues and cut very thin sections from a few micrometer up to 50 micrometer. Microtomes can be used for tissues that are frozen or also at room temperature, and they cut thicker section up to 100 micrometer. Vibratomes are used always used at room temperature, and they cut 6 sections above 100 micrometer. Okay, now that we know how we want to, which kind of tissue we want to work with, we know what we need to how we want to cut it, depending on what we have. So let's continue and see how we want to detect our protein of interest or proteins of interest. And as I said, there are two ways, either a chromogenic or fluorescence detection. So let's see what is the difference. Let's start with the chemical reaction. Where here we have our sample, and then we have our antibody, which has an enzyme which is bind to it, and we will add the substrate. In the substrate to the end, it's basically a color dye, also called the chromogenic dye. So then after incubating the sample with the antibody coupled to the enzyme, the substrate is added, and this will lead to a chemical reaction between the enzyme and the substrate. As a result, a precipitate will form near the antibody, making a color spot. After that, you are basically washing away the rest of the substrate. And the most commonly used enzyme are horseradish peroxidase or alkaline phosphatase. And for example, widely used substrate, it's DAB. The second method to detect your protein of interest is to it's the fluorescence method, in which case the second your antibody you will have a fluorophore which is attached to either directly to your primary antibody, or is it's attached to secondary antibodies. So these fluorophores, as we know, have the ability to absorb and emit light. And this actually, the fluorescent method is probably the most preferred method in the research environment, because it leads to higher contrast, higher spatial resolution, and higher sensitivity as such. Higher sensitivity, but it's also more expensive than the chemical method. But regardless of the detection method you use, if you use chromogenic or fluorescence, one can either, as I said, directly label the primary antibody, or use indirect detection by labeling the secondary antibody. So what are the advantages or disadvantages of using directly labeled or indirect labeled antibodies? So if we use directly labeled primary antibodies, of course, we will have a shorter experimental procedure. If we are doing multiplexing, then these antibodies are very useful, especially when we are sharing the same host species. And will also, then we can minimize the risk of non-specific signals due to the secondary antibody cross-reactivity. But of course, it has its disadvantages, because the signal will be weaker. We only have one fluorophore coupled to the antibody, and there are not too many available directly labeled primary antibodies, although now different companies are coming up to very short protocols, or you can do it actually yourself. You can couple your fluorophore to your antibodies. So it's getting better. Now, if you are using indirect labeling, it has its advantages. Of course, we will get a signal amplification because we will more secondary antibodies can bind to the primary. And of course, there is a much wider range on available labels for the secondary antibodies. But the disadvantage is that it takes, of course, longer time, longer experimental procedure. Well, I would say 1 extra hour is not the end of the world, but it's longer. It increased risks for non-specific signals due to secondary antibody binding, and multiplexing is quite limited because you will need to select an appropriate secondary antibody or combination of antibodies. So, chromogenic labeling versus fluorescent labeling. When should we choose what? Now, the chromogenic labeling always requires a color camera, with color cameras, which are less sensitive than the black and white cameras, but they are cheaper. When you are doing chromogenic labeling, one can label different antigens in the same sample using different chromogenic dyes. However, because all the colors are imaged at the same time by the color camera, they cannot overlap. So different colors can only be used with, then for example, we label different cell types or different cell components within a cell, like, I don't know, cytoplasm nucleus. Otherwise, you will not be able to distinguish. Another disadvantage is that that you are using chromogenic detection is that the contrast, sharpen, and resolution are lower than in fluorescence detection. The good thing is that if you are using chromogenic detection, then you won't have problems with the autofluorescence, but maybe sometimes with some endogenous tissue coloration, but that you don't have problems with autofluorescence is definitely a plus. Another advantage is that with the chromogenic method, besides being cheaper, is that the colors are preserved longer. Now, in the fluorescent labeling, you will have to use you are using a black and white cameras. Of course, you can image in color cameras too, but it's not. It's much better to use black and white cameras, which are more sensitive, and because you are able to label more molecules in the same sample, you will always be able to separate their localization because you label it with different fluorophores and you are excite them in different ways. So you are not restricted by where that you have to have different localization of your antigen. You will always get a better contrast in your sample, better resolution. But unfortunately, autofluorescence is a problem. And you will have to deal with it during your immunostaining. Also, you have a risk of bleed through between your fluorescent dyes, which of course we know by now that you will choose fluorescent dyes, so you can avoid as much. You will avoid bleed through. And another disadvantage is that the fluorescent dyes can fade during imaging and during storage. Now, how should you, of course, our goal is always to see more, to detect more, to perform immunostaining, and we want to detect more antigens, several antigens. So it's quite important to choose the correct primary and secondary antibodies. So here is a little example. We have three antigens. Let's say that antigen 1, it's recognized by an antibody which is raised in rabbit. So that means that the secondary antibody will bind to the FC part of the primary to the primary. So it has to be a rabbit a donkey anti-rabbit secondary. So it has to be against the host. Now, if we have the another antigen 2, the first primary antibody is a mouse anti-antigen 2. So again, your secondary has to your secondary antibody has to be raised in a species which is different. So in our case, it will be a donkey. It will be donkey anti-mouse. For the third antigen, we raised it with a primary goat anti-antigen 3. So the secondary will be a donkey anti-goat. So the secondary antibody must be raised against the host species of the primary antibody, and it has to be specific to the isotype class of the primary antibody, for example, IgG or IgM. Before starting your immunostaining, you have to you should think about what controls you should have you should perform together with your main sample. So there are three main type of control that you should always have next to your main sample. The no primary control, and where basically you emit the primary antibody, you only have a secondary, and this will tell you this control is needed to determine if your secondary antibody is binding specifically in your sample, giving you some false positive results. You will proceed with this sample exactly in the same way as with your main sample. And then we have autofluorescent control, where you again, as we know, we already heard before, some certain cells and tissue, and especially though which are very abundant in collagen, elastin, lipofuscin, brain tissues, usually liver, are quite autofluorescent, and quite often the autofluorescence signal is very specific. And if it's you don't pay attention, you would think, oh, this is a super duper, my signal is there, and when actually it's just autofluorescence from other molecules in the tissue. So you have to recognize and this kind of signals. So you should always have control a sample without absolutely nothing, no staining at all. But you should actually proceed with the same sample in the same way as you are performing your main sample. Then you have expression controls. But in some cases, it is possible to use negative and positive control, which is basically a negative control can be a knockdown or a knockout. And so you eliminated the level of your expression protein, of your antigen. And then you can use this negative control to see if your antigen it's binded or not. So you should see no staining basically in that sample. Or you can have a positive control, so you have an overexpression of your antigen levels, especially on you can overexpress. It's easier done in cell lines, but you can also use some tissues where you know that this particular antigen is present in high levels. So you should see a very proper staining in these tissues, so you know that everything works as it should. And then we have isotype controls. These are quite routinely used in flow cytometry, but there are situations that when we should also use these controls in immunofluorescence too. And these particular cases involve if the tissue is involved in immunity, which will likely make it susceptible that it have FC receptors present. So you want to know if your antibody will bind there. If the tissue is highly infiltrated with immune cells, again, they will present FC receptors. If the primary antibody is raised in the same species as your mouse, for example, then you will have to use these isotype controls. As I said, the control samples must be processed in exactly the same way as your main sample. So now that we will have a little break, a 5 minutes break, and then we will continue with the next part, how we are choosing our antibodies and what is a good antibody. Okay? Just continue. Oh, okay. So how do we choose an antibody? Can everyone hear me?
Yes.
Yes.
So basically, the most common way to get hold of your antibodies to search the literature, but unfortunately, you might not be able to use the antibody mentioned in an article, or you might not obtain the same results than using it. And in a survey from 2018 showed that more than 50% of all cited antibodies could not be identified or purchased due to poor material and methods reporting, the change of the product name or number or discontinuation, and so on and so forth. So that's not very funny that we actually are looking, you know, I want to use this. It shows good results, but you cannot find it. Of course, the second, another way is, of course, you go directly to the companies who are selling the antibodies. And then, of course, you find what you want, but you should be very critical, and you should see that then you look at and you buy that antibody, that it has a complete data sheet which is available, and it contains as much data possible about that particular antibody. Western blot, stainings, images, usage recommendation. Sometimes they even have a protocol, but fair enough if they don't. But nevertheless, and some validation data. So missing this kind of information, like, I don't know, Western blot, basically it says that they didn't even look what is the molecular weight of this antibody. If you're missing an image, you should ask yourself, did they actually try it, if this is what you want to use it for? But there are other ways on how to find an antibody. You can go online and to different antibody databases, so-called antibody portals. For example, the antibody portal or Antibodypedia They have open access and provide information about the performance of antibody in specific applications, not like Western blot, immunoprecipitation, immunostaining, and you can have which are all based on experimental results and publication, and they are verified. Another portal is the antibodies online. Here, independent labs can do validation studies, generally at the vendor expense. And those antibodies which pass the validation test will are so-called independent validation badge. And we also have the antibody registry, which usually assigns an RRID number, so-called research resource identifier to the antibody. And then you can search in their database or find this number in a publication. You can go back to this registry and look up your antibody using this number, and you will get a lot of information, tips, who's selling it, pluses and minuses, and so on and so forth. So go out there and look a little bit more, not only get what you look at for where to get in your publication, because double check everything before you buy an antibody. But now let's say that you decided, I will buy this and I want this antibody. So what kind of information should a vendor provide? And as I said, the data sheet is a must. It should be always provided with your antibody. But of course, what is on that data sheet also makes a difference. So you should always be provided with a clone number, commercial product number, antibody ID. And if you are lucky, you will also get an RRID number. What kind of antibody it is? It's a polyclonal, a monoclonal, recombinant, and of course, isotype class and subclass. A clear definition of the immunogen. What was this antibody raised against? You know, a peptide, protein fragment, or the full-length protein with the 3D structure. And because this will allow you to decide, knowing the immunogen, if this antibody will be specific enough or not for your application. Cross reactivity is also very important, and basically it will, by knowing cross reactivity, it will tell you, can I use this antibody for my application? And the more cross reactivity data they provide and they show, the better you can evaluate its usability in your application. And that also means, probably, I would say that more company worked on validating this, validated this antibody. Concentration of antibody, additive, and buffers. Of course, if you do not get a concentration, is not provided, you will have a very hard time to optimize your protocol. And if you don't know what additives or buffer they are not specified, that maybe that will prevent you, for example, for conjugated your antibody with fluorophores if you want to do a direct labeling. Antibody affinity. Does this antibody have a high or low affinity? It's a huge difference between a weakly binding and a high affinity binding antibodies, because I would say a low affinity binding antibody would lead to a weak signal, a specific background, low reproducibility, and most of all, it will cost you a lot, because you will, of course, instinctively add more and more, hoping that you will see more. How you should storage your antibody. of course, it's a must. Considering how the high cost of many antibodies, stability is very important. So recommendation about storage conditions is a must. Now, the application field. Should this antibody be used in Western blot, immunostaining, laser? It's quite an important information. But if, let's say, this was shown to work, for example, in a Western blot, that doesn't necessarily mean it won't work for immunostaining. It could also only mean that they didn't try it there. So if you really want this antibody, you should also do your validation yourself and see if it works or not. But it takes time, of course, to do all the validations. But if you don't find anything, or no Western blot is shown, no immunostaining, no specific immunoprecipitation data or other data, then please, you have two choices. Either you don't buy your antibody or you do all these validations yourself. And in the best cases, the companies will provide you with a protocol, and they will show how they validate the data. Here also, still you will have to ask yourself, was this validation done in cells or in tissues? If it was a tissue, it was fresh frozen or paraffin embedded, or and so on. And if you don't find, or you don't only find half of the information, then you have to do the validation yourself. Okay. So let's move on and let's see how we characterize an antibody. What is a good antibody? A good antibody should fill the following criteria. It should be specific, so which is the most critical issue. So it should only detect the target antigen. It should be sensitive. It should detect low amounts of the antigen, and it should be should have high reproducibility. So basically, same level of should show the same level of specificity and sensitivity across batches and time. Okay, so how can we see if an antibody is or how can we validate our antibody for immunostaining and check its specificity, sensitivity, and reproducibility? So let's start with the Western blot. Oops, something is there. With specificity, Western blots are widely used to determine the specificity of an antibody. They are easy and cheap to perform, and they will show you that the antibody binds to the antigen, and it has the expected size. But the Western blot in the datasheet is only informing you that the antibody is able to bind the denatured protein, and that's very important to keep in mind, because, for example, if your antibody was raised against a folded protein, of course, the Western blot cannot be used because the antibody will not recognize it. And this is a case, for example, for activated integrin. Also, then the Western blots can be performed on purified recombinant proteins or sample extracts. And if it's against the purified recombinant protein, it should only show one band. If it is again different cell extracts, ideally it should only show one band or several bands if the protein have isoforms or some post-translational modification, as you can see here. But for example, in this blot, we can see that two cell lines also have some extra additional bands, which could make me think that there is some specific binding in these cells. But of course, the manufacturer can obviously not test all possible type of samples. And of course, they probably won't show a Western blot using exactly the cells you are working with, and even more if you are working with tissue, that they will present some data from extracts from a tissue. So this is something that then you have to validate yourself. If it can also happen to be vice versa, that you have some data from tissue extracts, but you are working with cells, then you can actually do something about it. You are taking your cells, you pellet them, you have your pellet, and you handle that pellet like it should be a tissue, a piece of tissue. So you will froze them or paraffin embed it if you want, and then you will cut and perform all the steps exactly like it should be a tissue, and you will see what results you get. And again, an antibody might work in Western blot, but not immunostaining, and the reverse is also it's also true. And again, another disadvantage of Western blots are that they do not show the localization of the antigen. So if you do, you need to use it in immunostaining, then you have to do the validation yourself. Now, how are we dealing with specificity? Here we have overexpression, knockouts, and knockdowns that can be done in cells or cell pellets or tissues. And this will allow us to increase or decrease the level of the protein of interest in cells of tissue. And we will check if the immunostaining or the Western blot varies as expected. And is the gold standard for demonstrating specificity, these three methods. And by increasing or decreasing the levels, you will see if you get what you expect. But it's if you have to see if your antibody works in a tissue, or it will be much harder to have any overexpressed or knockdowns or knockout. So you can do this, as I explained in my previous slide, by taking your protein of interest, pellet down, and process them as tissue. The minuses of this approach is that there are very limited numbers of knockdowns and knockouts available in animal models. Producing transgenic animals takes time and money, so yeah, you might use cell lines. Some genes cannot be knocked out as they will lead to low cell viability, and knockdowns are generally transient, rarely 100% effective. So you need controls like real-time PCR or Western blot to validate them. Affinity, basically how strongly does the antibody bind to your antigen. It's also a very important piece of information. And in order to be, you will have to this information is rarely provided on datasheets, so it's not always possible to compare the antibodies in that way. An antibody will always bind to many proteins, but of course it should bind to a much higher affinity to the target antigen, and through washes and stringent incubation conditions will ensure that it binds to its specific antigen. And by stringent conditions, we mean that we make it much harder for the antibody to bind, which sounds counterintuitive. But with that, we'll ensure a specific signal and a high contrast. And how can we increase the stringency? We can lower the concentration of the primary antibody quite a lot. We incubate with the primary antibody at low temperature, usually at 4 degrees, and for a longer time. Also, we can do dilution series, and we especially when you are using the primary antibody for the first time. You should always do a dilution series. You start with exactly what is written in the protocol, and then you just dilute by going big steps. And you will find the place where actually you will get the best staining without any background all around. Antibody stability, which is also important, as I said, a piece of information that the antibody should stable over time under the expiration date. And to preserve this for an antibody, aliquot as soon as you receive it, keep a working stock, write the date of the first use on the vial, and most important, store the antibody as recommended. If the antibody expires and you still use it, keep a critical mind, and you are looking at your images. Now, reproducibility between experiment, then using the same batch or between batches. If you want to check the reproducibility on your current batch, run the same immunostaining protocols on 3 different ways and compare the results that you get. Obviously, you should get the same. If you want to compare reproducibility between batches, then you should, for example, your batch that you are using now, your antibodies, it's almost running out. Please order the new one in due time, so you can compare these the two batches and see how well the new batch is performing. And if you feel that, if you see that it does not, either you don't get any staining, or you have to use a lot of the antibody of the new batch, complain to the company, and so they can send you a new batch of the antibody. And of course, you have a higher risk for to have a higher risk for lower reproducibility than you are using polyclonal antibodies. Of course, you should not forget to validate your secondary antibodies, which must satisfy the same standards as primary antibody in the sense of specificity, affinity, stability, and reproducibility. And here you can see an example of an antibody, a secondary antibody to different batches. You can obviously see there is the same antibody to different batches, and there's a difference between them. Always have a control sample, for the secondary, also for the secondary antibodies, so you can look at the cross reactivity with endogenous IgGs or serum IgGs. But that can be also easily solved if you use the correct blocking. Always, then you buy your secondary antibodies, demand the full spectrum of the fluorophore bound to the secondary antibody, not only the maximum of its excitation and emission. Sometimes be aware that the same antibody may be sold under different names by the different companies. So that will mean that you will waste a lot of time of testing different antibodies, although they are actually the same. And the sign that you are that will tell you that you are dealing with the same antibody is the same formulation in their data sheet, the same images of the Western blot or immunostaining, same vial size, same recommended protocol, that basic same reference list, that basically tells you that the other company is actually selling the same thing that that you had before. It's not new antibody, is just packed it in with a different vial and give it a different name. So if you think that you buy something new, well, something that might work better, then you actually buy the same thing, and it's a waste of time and money. Okay, so now we know also about the antibody. We decided on all the other steps before. Now we have to pay attention to a few things in the immunostaining steps. And we already talked about fixation. And of course, the main goal is to preserve the cellular morphology, integrity, and structures. And depending on what you are using, cross-linking agents or organic solvents, they will work differently because they interact differently with the sample. The cross-linking agents will form bridges, and their role is, of course, it will preserve quite nicely the cells and tissue structure, but they require permeabilization after using. Usually this fixation is performed at room temperature. It takes time to adjust the time you need for fixation, especially for the fixed tissues, which also mean that you have to be very careful not to overfix, because that will introduce artifacts and mask the epitopes, or underfix, to have too short, which will lead to a degradation of your target. The other type of fixative are the organic solvents, methanol, acetone, ethanol, and they can also be used together, which act in a totally different way. They dissolve lipids, dehydrate the sample, and precipitate proteins. They are much harsher on the cells and tissues. If you use this, you don't need to permobilize. This process is usually performed on ice, and it's quite short. Always check the applications note and the customized protocols on the data source of the antibody you use to know exactly which fixative to use, because some will definitely not work if you, for example, have methanol in your fixative, or with methadolol. And then I say methanol in your fixative, I mean the formaldehyde, which is stabilized sometimes with the methanol. Permeabilization, here again, is not a trivial. Don't use always the same Triton X100, because that's what I heard in the lab that we are using. Because what the role of the permeabilization is to make holes in the membrane to allow an antibody penetration, but depending on the type of that you are using, you have detergent or organic solvents. Within the class of the detergent, you have the mild ones, like saponin, digitonin, twin 20. They will only permeabilize some type of membranes, especially those which are containing cholesterol. So if you use saponin or the digitonin on twin 20, the nuclear membrane will not be permeabilized. Then this process is reversible, so you have to have it in the solutions all the time. Then you have the more harsh detergent, like Triton X100 and MP40, that will permeabilize all membranes and all membranes. And you have to adjust also here is quite critical to adjust the concentration, because if you are interested in the nuclear membrane, you will always have much higher concentration if you want, for example, to permeabilize the Golgi membrane or the ER. We have the organic solvents, the most classical methanol and acetone. They dissolve lipids, so they make holes, and quite big holes in the membrane. That's why they already perform in ice. We have to be careful because they usually damage microtubules and some other organelles. Always use the permeabilization agent suitable for your scientific question. Again, just not follow a protocol because it's there. think a little bit over what you want to actually look at and where that protein of interest is. Always adjust the concentration and time depending on the thickness of the tissue. If you are using organic solvents, make sure your antibody is compatible with using it. Now, antigen retrieval. Why do we do antigen retrieval? Because during the fixation step, some crosslinks are formed and they will mask the epitope, so your antibody will not be able to bind to it. So when we perform antigen retrieval, we break the crosslinks which are generated by this fixation, so we can mask the epitopes. So now the antibody can bind. They are always performed before the staining, and there are two methods: the heat induce or enzyme induce. The heat induce is the mostly used method. Usually you need a high temperature, which basically water-based microwaves, pressure cookers. This method is quite sensitive to pH, temperature, and the buffer composition. So you have to optimize these things depending on your tissue fixative and antigen. And here you have, again, I mentioned before, you might have a problem that your tissue will detach from the cover slip. So if you manage to put it there and you want to try that, you should try. I suggest that you incubate them horizontally in the buffer, and then you hopefully they will not slide down. We have the enzyme-induced antigen retrieval method. Is less used because it has a much higher risk of tissue damaging the tissue morphology and the antigen, your antigen. It is more tricky to adjust the concentration of your enzyme, which usually use protein A, trypsin, or pepsin. So all these, the temperature has to be very, very careful optimized. Do you always need antigen retriever? No, it's not written that you always see. Most of the case with the paraffin embedded section, they say you need, but sometimes you don't have to do this harsh antigen retrieval method. For example, you could dilute the antibody at a slightly different pH or cation concentration, and it might help. You can have different dilution and incubation times. You can choose a polyclonal antibody instead of a monoclonal, so you will have different, more epitopes available to bind. And maybe you don't have to do the harsh antigen retrieval. And here is just an example where you, by changing the concentration and the pH in the buffer between different antigen retrieval buffers, you can see that how the staining it's changed from pH 3 you already see, and then for example for the progesterone, and but nothing at pH 4, 5, and 6. And then you can see again, you use a different buffer. Again, you see two totally different stainings. And that because the buffers work, then the pH work differently. The blocking, we have different blocking agents, and is to compete with a specific binding of the antibody, and is done before adding the primary antibody. We have different blocking agents, normal serum, which should be from the host of the labeled antibody. So if you have antibody from different species, it might be necessary to mix the blocking sera from the species of the all secondary antibodies. You can have protein buffers, which are the classical we are using, BSA, non-fat milk, casein. But here you have to be aware that if they might be contaminated with bovine IgG, which will increase the risk of the background if you are using an anti-bovine, anti-goat, anti-sheep antibody, because it might react. You can have FAB fragments, as I mentioned also before, which are used to block endogenous immunoglobulins in tissue section or on cell surface. Then the primary antibody is raised in the same species as the sample. And here I have an example. Then we have a mouse tissue, and we want to use a mouse antibody, which is against antigen X on the tissue. But we have endogenous IgGs, then we will use the FAB fragment to block this endogenous IgG, and then we can add our primary and the secondary, the primary mouse antibody, and then our secondary antibody without risking binding to the endogenous IgG. Of course, the blocking should be also valid for chromogenic labeling, because quite a lot of tissue, they have this enzymatic activity. So you have to perform a peroxidase blocking if you are using the HRP detection. And in this case, you will use hydrogen peroxide. Or you can, it should be, you can use it either before or after, depending how your, what is your detection that you are doing. And you can also, you have to also do the same if you are using an alkylase phosphatase block, when you are using alkylase phosphatase detection, because a lot of the tissues, some tissues have quite a lot of alkaline phosphatase. And of course, there are several available. The levabisol is the one that is most widely used, and is usually added together with the chromogenic substrate. You have to, if you are using biotin detection system, biotin also needs to be blocked, and because it's especially present in many tissues, especially kidney, liver, and brain. And you will need pre-incubation of this tissue with avidin, streptavidin, and again, biotin to block all these binding sites. Washing steps are very important, and they should be added in your immunostaining protocol, and you should wash a lot, but gentle. Then the common buffer PBS or PBST, depending on what you are, if you are doing fluorescence or chromogenic staining, and or if you are using if you are immunostaining phosphorylated against phosphorylated epitopes, you should use the TBS or TBS T, or when you are using chromogenic labeling with alkaline phosphatase. And all these things are very clearly specified in the protocols, but sometimes you have a tendency to hop over some washing step or just do them quickly. And that's not good, because it will increase your background quite a lot. In conclusion, always check that the antibody you want to use is validated for the immunostaining, and in any case, validate the antibody yourself for your own sample. Always start by carefully reading the recommendation and protocols provided in the datasheet. And if you find that the information is not sufficient, write to manufacturer and demand that they provide it. If there is no protocol, that means that they probably don't even have tried anything. So then you should think twice if you use or you return the product. Always follow the given protocol: fixation, blocking, washing, and dilution. Then add extra dilution to test how low you can go, so you can decrease the background. Test new antibody of batches and compare to the previous one. If it's not good, demand a new batch. Do not forget about controls. Always have the controls then you are starting a new antibody. Do it once, do it right. Be critical with the obtained results. We are always usually quite happy. I got the signal, but look carefully. Is it in the correct place? It's in the correct context. Check the literature, and of course compare with the data sheet if any it was available there. And remember, there is no such thing as a perfect protocol. Most protocols need optimization, and it's up to you to do that optimization. Thank you.